Moving on from the stomach, we come to the small intestine. The small intestine, despite its name, is actually the longest region of the digestive tract. It's called the small intestine because it's smaller in diameter than the large intestine, although it is much longer. It's about 20 feet long in a living person and can be up to half again as long when dead, so up to about 30 feet as long when dead and the smooth muscle is fully relaxed. The small intestine runs from the pyloric sphincter between the stomach and the duodenum to the ileocecal valve between the small intestine and the large intestine. There are three regions of the small intestine. The first, the short duodenum, takes this kind of C-shaped curve. Then you have the jejunum, and after that you have the ileum. There are some histological changes as you move along from the duodenum to jejunum to ileum. There are more mucus glands in the duodenum and smaller villi, the finger-like projections. There are mini villi in the jejunum, and the Peyer's patches, those clusters of lymphoid tissue, those you find in the ileum. The main functions of the small intestine are digestion and absorption. Digestion is nearly completed here, and almost all absorption occurs here. This is also where juices from the pancreas and bile from the gallbladder enter into the digestive tract. To see what the small intestine actually looks like, we're looking at cadaver images. The small intestine are these kind of worm-like structures framed within the large intestine. And then you can see the mesenteries, those folds of peritoneum, holding the small intestines in place here. Since almost all absorption happens in the small intestine is highly modified in order to absolutely maximize the efficiency of absorption. You have these large circular folds called the plici circularis. These are deep folds involving both the mucosa and the submucosa. They're permanent, so unlike the rugae in the stomach, they don't disappear, they're permanent. And these circular folds force chyme to spiral through the lumen, slowing it down and bringing that chyme into contact with the epithelium. You also have villi and microvilli, and both of these really increase the surface area for absorption. To think about the role of surface area in absorption, think about an ordinary kitchen sponge and a pool of water that needs to be absorbed. Which sponge is going to absorb that water more quickly? Which sponge is going to absorb that water more efficiently? I bet you picked this one, right? More surface area is in contact with the water, so this sponge is going to absorb a lot more water a lot more quickly. The same principle holds true with absorption in the small intestine. More surface area gives you more efficient absorption. The villi are finger-like projections of the mucosa that increase the surface area coming into contact with the chyme. You also have a dense capillary bed and a lacteal lymphatic vessel in the center of each villus, so that when nutrients, which are being formed through digestion in the lumen, they can cross straight across the epithelium into the capillary or into the lacteal. Microvilli are microscopic extensions on the surface of epithelial cells. Okay, so they're microvilli, they're like really, really small little projections, and they're on the surface of individual epithelial cells. This is a zoomed in version of that. So to kind of try to sketch out what that looks like, if you have a single villus, villi is the plural form, villus is the singular form, each cell on that villus has microvilli. So villus is the finger-like projection. Microvilli are the microscopic projections on the surface of the intestinal epithelial cells. They don't move, they just increase the surface area. Here's a look at the microvilli in an electron micrograph. You can see this is a single cell. There's the border of the cell. It's the other border. And here are the microvilli. These are stiff projections. They give the they look almost like brush bristles on the surface of the epithelial cells. So sometimes the microvilli are referred to as the brush border. And when we talk about digestive enzymes in the small intestine, you'll hear reference to brush border enzymes, because there are enzymes sitting on these microvilli that finish carbohydrate and protein digestion. So now that we've looked at the microvilli, I'll give you a look at the villi too. These are finger-like projections. These are scanning electron micrographs comparing the villi of the small intestine with the gastric pits of the stomach. See how the villi project out into the lumen, and the gastric pits sink in away from the lumen. With these folds, the entire surface area of the small intestine, the area that's available for absorption, is about the size of a tennis court, all packaged up in your abdomen. At the base of the villi, you have intestinal crypts, and this is where you find the hormone-secreting cells, the enteroendocrine cells of the small intestine. These are basically tubular glands that sink in below the surface of the small intestine. They secrete watery mucus. There also are stem cells at the base of the crypt that replace the mucosal cells every 2 to 4 days. So these cells up here at the peak of the villus, of each villus, are coming from stem cells located at the base of the intestinal crypt. These cells down here are dividing and remaking the epithelium of the small intestine. You also have immune cells here that help protect against foodborne pathogens. You have Paneth cells. Paneth cells release defensins and lysozymes, so antimicrobial molecules. Intraepithelial lymphocytes are lymphocytes that definitely play a role in fighting against foodborne illness, and there's an active area of research that thinks that they may have a role in controlling the immune response as well, making sure that your immune system doesn't overreact to your normal microbes found in the digestive tract. The enteroendocrine cells here produce gastrin, cholecystokinin, and secretin. Gastrin stimulates the stomach to produce hydrochloric acid and pepsin. Secretin stimulates the pancreas to secrete bicarbonate into its juices to neutralize the acidity of contents coming from the stomach. Cholecystokinin, CCK for short, acts on the gallbladder, stimulating it to contract and release bile into the intestine. It acts on the pancreas, stimulating the release of pancreatic juices into the small intestine. And it actually gives a satiety signal to the brain, so helps contribute to that sense of fullness. Okay, after reviewing this video, you should be able to identify the regions of the small intestine, talk about how the small intestine is specialized for absorption, so the villi, the microvilli, and the circular folds, talk about the digestive activities, the digestion and absorption that happen in the small intestine, and discuss the functions of these cells found in the intestinal crypts.